Bismillahirrahmanirrahim. Here is Mobeen Naz. I'm from MK Preparations, and today we'll discuss about biological molecules. So, dear students and candidates, this topic is very much important for the preparation of different competitive exams like Punjab Public Service Commission (PPSC), Khyber Pakhtunkhwa Public Service Commission (KPPSC), Sindh Public Service Commission (SPSC), Balochistan Public Service Commission (BPSC), and Federal Public Service Commission (FPSC). So this topic is very much important for the students as well who are going to secure admission or who want to secure admission in any medical university or any department that is related to allied health sciences. So the material coded in these slides have been taken from different source books, which includes the FSC Biology, Federal Biology, Zoology Miller and Harley 5th and 10th edition, Hickman Zoology, Campbell Biology, and Riazul Haq Rame book. Okay, so first of all, we'll discuss about the contents that we are going to discuss in these slides or the topics. Okay, so the first one is the general introduction of biochemistry. Then we'll discuss about the carbohydrates, then carbohydrates classification, and then lipids functions and classification, then proteins, and then about the protein structure, functions, and classification. Then nucleic acids, which includes the DNA and RNA, and enzymes, enzyme structure, functions, and factors affecting the enzyme activity. Okay, so first of all, now we'll discuss about the biochemistry, that what is biochemistry? Okay, ji. So biochemistry is a branch of biology which deals with the study of chemical components and the chemical processes in living organism. तो biochemistry जो है ये biology की एक ऐसी branch है जिसमें हम chemical components यानी अपनी body की living things की जो chemical composition है उसको discuss करेंगे and इस chemical composition के अलावा हम living cells में जितने भी metabolic reactions हो रहे होते हैं, ठीक है, उन सब चीजों को discuss करते हैं. So biochemistry is essential for understanding the anatomy and physiology. So biochemistry जो है एक बहुत important branch है in which we get the knowledge about the structure and the function, means about the anatomy and about the physiology of the living things. For example, the photosynthesis, respiration, digestion, and muscle contraction. All of these are the topics that are related to physiology. Means the function of the living things. So we'll discuss about these processes, okay? And all the chemical reactions taking place within our cell are collectively known as metabolism. Means एक cell में जितने भी metabolic reactions हो रहे होंगे collectively उन्हें हम जितने भी reactions हो रहे होते हैं, ठीक है, chemical reactions हो रहे हैं, उन सब chemical reactions में something is going to be synthesized or something is going to be broken down. So ऐसे reactions जो हैं, these are known as metabolism. And metabolic processes are further divided into two types, the anabolic and the catabolic processes. Okay. So first of all, we'll discuss that what is anabolic reaction. So these are the reactions in which simpler substances are going to combine to form the complex substances. Okay. And the example of this anabolic reaction is the photosynthesis, in which we know that carbon dioxide will combine with the water to synthesize the glucose. Okay, so photosynthesis is a type of anabolic reaction. So in the anabolic reaction, when something is going to be synthesized, so we need energy. Like just for the photosynthesis, if we're talking about the photosynthesis, then the demand of energy will be fulfilled by the sunlight. Okay. Then is the catabolic reaction. So catabolic reactions are those reactions that involve the breakdown of the complex molecules into the simpler ones. Okay. So in this, the complex molecules will be broken down into simpler molecules. The example of the catabolic reactions include the respiration, okay, and digestion. So in respiration, if we're talking about respiration, so in respiration, the glucose will be broken down in the presence of or in the absence of oxygen into the subunits, okay. So glucose will be broken down, and in this, a lot of energy will be released. Some energy will be utilized by the cells directly, and some will be stored in the form of ATP, okay. So the catabolic reactions are those in which the energy is going to be released. Okay जी, so ये हमारे पास एक जनरल सा इंट्रोडक्शन था कि बायोकेमिस्ट्री में हम केमिकल कंपोजिशन और केमिकल प्रोसेसेस को स्टडी करते हैं, एंड दीज केमिकल प्रोसेसेस आर इन्वॉल्व्ड इन द मेटाबॉलिक रिएक्शंस, एंड the metabolism is further divided into catabolism and anabolism. तो इन दोनों का जो difference है, that is very much important, okay. Now we'll discuss about the organic and the inorganic molecules. So the organic molecules are those compounds which are composed of carbon and hydrogen together in a molecule. And we have divided these organic compounds or molecules into two types. The larger one that are known as the macromolecules, and the example of these macromolecules is cellulose, fats and proteins, okay. तो ये macromolecules हैं, इनका size, इनका weight, molecular weight इनका काफी ज्यादा होता है. और उसके बाद है हमारे पास small organic molecules, so these are known as micromolecules, and the examples of the micromolecules include the glucose, amino acids and fatty acids. So you can see here glucose is the subunit of the cellulose, amino acids are the subunits of the proteins, and fatty acids are the subunits of the fats. So definitely they are having a less molecular weight, okay. So these are small organic molecules. Now the inorganic molecules. So inorganic molecules are those in which the carbon and hydrogen are not present together in a molecule, okay. Means carbon और hydrogen अलग अलग तो हो सकते हैं, like carbon dioxide or like water. तो ये दोनों examples हैं हमारे पास inorganic molecules की. But in both of these examples, you are unable to see the carbon and hydrogen together in one molecule. But if we're talking about the glucose, cellulose, fats, proteins, amino acids, fatty acids, so you will observe the carbon and hydrogen together in their formulas or in their molecular formulas, okay? Now, we'll discuss about the carbohydrates in detail, okay? So, most important जो हमें यहाँ पे discuss करने हैं biological molecules, these are carbohydrates, lipids, proteins and enzymes, okay? तो one by one हम इनको discuss करते हैं जी. First of all, the carbohydrates. Okay. So the word carbohydrate literally means hydrated carbons. Okay. And the composition of the carbohydrates that they are made up of carbon, hydrogen, and oxygen. And the ratio of hydrogen and oxygen is just same like the water. As you can see here, the general formula of the all carbohydrates, CX and H2O, Y. So H2O means the ratio of the hydrogen and oxygen is exactly same as of water, okay. अगर हम अब प्रॉपरली डिफाइन करें, केमिकली डिफाइन करें, इसका जो लिटरल मीनिंग है या आप कह लें कि जो लफ्जी मतलब है वो तो है हाइड्रेटेड कार्बन, ओके. लेकिन अगर आप इसको केमिकली डिफाइन करें तो किस तरीके से कर सकते हैं, दैट दीज आर पॉलीहाइड्रोक्सी एल्डिहाइड्स और कीटोन्स, ओके. पॉलीहाइड्रोक्सी मीन्स इनके पास बहुत ज्यादा हाइड्रोक्सिल आइंस प्रेजेंट हैं विद द फंक्शनल ग्रुप्स एल्डिहाइड्स और कीटोन्स, ओके. तो इनके साथ ये एल्डिहाइड या कीटोनिक ग्रुप अटैच होता है. Or these are complex substances which on hydrolysis yield the polyhydroxy aldehyde or ketone subunits. या हम इन्हें कह सकते हैं कि वो substances हैं जिनकी अगर हाइड्रोलिसिस हो, इनकी अगर ब्रेकडाउन हो in the presence of water, so they will yield the polyhydroxyaldehyde or ketonic subunits. Now here's the definition of the hydrolysis, that it's a breakdown of the larger molecules into the smaller ones utilizing the water molecules, okay. Now, the general formula of the carbohydrates is CxH2O whole Y, and X is the whole number from 3 to many thousands, means के जो X का जो number है यहाँ पे 3 to many thousand तक हो सकता है, यानी कार्बन के नंबर की बात हो रही है, and Y may be the same or different whole number. Now, the carbohydrates are found in all the organisms and in almost all parts of the cell. Okay. Examples of the carbohydrates includes cellulose. So the cellulose is present in the wood and cotton and paper. Okay. Then next example is the starch. So starch is present in cereals and root tubers and cane sugar and milk sugar. Okay. So these are all the examples of the carbohydrates. So in this slide, we studied about the composition, the literal meaning. We defined the carbohydrates chemically. We studied about their general formula and their examples. Okay, Ji. Now, the functions of the carbohydrates. So, carbohydrates play the structural and the functional roles. Structural role because they are the part of the membranes, and functional roles because they are providing the energy. These are the sources of energy, actually. So, they are the main sources of energy in the cell. Okay, means the breakdown of carbohydrates would release a lot of energy. Main constituents of the cell wall in the plants and microorganisms. So means they are involved in the composition of the plant and the microorganisms of cell wall. So because they are present in these structures in the walls of the plants and the microorganisms, so they are performing the structural role. Similarly, they are the source of energy, so they are performing the functional role here. Okay, जी. So now, what are the sources of carbohydrates? Actually, means हम carbohydrates को कहाँ से obtain कर सकते हैं? So these are the green plants. Okay, green plants are doing the photosynthesis. So in case of photosynthesis, we'll get the glucose, and glucose molecules will then combine, and they will be converted into the different types of the carbohydrates. Okay. Now, they are the primary products of the photosynthesis, and carbohydrates in the cell combine with the proteins and lipids to be converted into the conjugated molecules that are known as the glycoproteins and glycolipids, respectively. Means when carbohydrates are going to combine with proteins, they will be converted into glycoproteins, and when these are going to combine with lipids, they will be converted into glycolipids. Okay. So the glycoproteins and the glycolipids, both of these also have the structural role in the extracellular matrix of the animals and the bacterial cell wall. Means the glycoproteins and glycolipids both are the components of the cell walls of animals and bacterial cells. Okay. And both of these are the components of biological membranes as well. So all of these are the functions of the carbohydrates. Now we are moving towards the classification of the carbohydrates. Okay, ji. So, carbohydrates are also called the saccharides. This word has been derived from the green word saccharon, which means sugar. Okay. तो इसी वजह से हम जो है ना वो carbohydrates को sugars भी कहते हैं. So they are classified into three groups: the monosaccharides, oligosaccharides, and polysaccharides. अब हम one by one इनकी details को देखते हैं. So first of all, we'll discuss about the monosaccharides. Okay, ji. So mono means one and saccharide means one sugar. So chemically, they are either polyhydroxyaldehydes or ketones. We can define the monosaccharides just like the carbohydrates generally. And these are some characteristics of these monosaccharides. That these are simplest, most sugars. Okay, and they are sweet in taste, and they are easily soluble in water. They cannot be hydrolyzed into much simpler sugars. Okay. All the carbon atoms in a monosaccharide except the one have a hydroxyl group. Means in monosaccharides, all the carbon atoms will have the hydroxyl groups except the one. And the one that is not having the hydroxyl group will definitely have the functional group, either aldehyde or the keto group. Okay. तो मतलब जितने भी हमारे पास मोनोसेक्राइड्स में कार्बन एटम्स होंगे सबके साथ हाइड्रोक्सिल ग्रुप होगा एक्सेप्ट दी वन, और जिसके साथ नहीं होगा इट्स गोइंग टू हैव द aldehyde group or the keto group. Okay. So the sugar with the aldehyde group is known as the aldo sugar, and the sugar with the keto group is known as the keto sugar. Okay. So the general formula of the monosaccharides is CH2O whole N. Okay. Now, we'll discuss about the classification of monosaccharides. So the classification of monosaccharides on the basis of a number of the carbon atoms. So in nature, monosaccharides have been divided into the different categories on the basis of number of carbon atoms. So if there are three carbon atoms, then these are trioses. Okay. If there are four carbon atoms, so these are tetroses. If they are having five carbon atoms, so these are pentoses. With six carbons, these are hexoses, and with seven carbons, these are heptoses. Okay, so the range of carbon is just from 3 to 7 carbon atoms. Okay, ji. Now we'll discuss about their formulas as well as we'll have a look on their structures. Okay. Monosaccharides, the two trioses mentioned here are the intermediates in respiration and photosynthesis. So what are these two trioses? As you can see here, these are the two trioses, the glyceraldehyde and the dihydroxyacetone. So just have a look on their formulas or their functional group. So you can see here, this one is the aldehyde group. So glyceraldehyde is an aldo sugar and it's a triose sugar. Similarly, dihydroxyacetone, it's a triose sugar, but you can see here it's having the keto group, so that's why it's a keto sugar. Okay, and both of these are intermediates in the respiration and photosynthesis. Tetroses are rare in nature and occur in some bacteria only. Pentoses and hexoses are most common. Okay, as you can see here, the 1, 2, 3, 4. This ribose is a 5 carbon sugar, and glucose, galactose, fructose. These are 6 carbon sugars. Okay. Now we'll differentiate these sugars on the basis of their functional groups. So most of the monosaccharides form a ring structure when in solution. So you can see here, ribose has formed a ring structure that is known as ribofuranose, and glucose has formed a ring structure that is known as glucopyranose. Okay. So first of all, we need to get to know about the ribulose. That ribulose is 1, 2, 3, 4, 5. It's a 5 carbon sugar. It's a pentose sugar. Okay, and this ribulose is a keto sugar. But ribose is also 5 carbon, but you can see here it's having the aldehyde group, so it's an aldo sugar. So glucose and the galactose, both of these are 6 carbon, means these are hexoses and aldo sugars. But fructose is also 6 carbon, but it's not an aldo, but it's a keto sugar. Okay. So we were talking about the ring structures of these sugars. So when ribose will form the five-cornered ring, it will be known as ribofuranose. So all the rings that are furanose, it means that they are having five corners. Okay, and those that are pyranose, it means that they are having 6 corners. So ribose is ribofuranose, 5 carbon ring, and glucose is glucopyranose, a 6 cornered ring. Okay. Now we'll discuss about the most common monosaccharide, that is glucose. Okay, so glucose in free state, the glucose is present in all the fruits and the most abundant in grapes, figs, and dates, and in our blood that is containing 0.08% glucose. Okay, but in combined form, it is present in the form of disaccharides or polysaccharides. Disaccharides means two glucose molecules are being joined together, and polysaccharides means many glucose molecules are being joined together. Okay, so starch, cellulose, and glycogen, they yield glucose on complete hydrolysis. So these three are the examples of the polysaccharides. Okay, glucose is naturally produced in green plants, and it can take carbon dioxide from the air and water from the soil to synthesize the glucose by the process of photosynthesis. And during this reaction, energy will be utilized from the sunlight. And this is why the process is called the photosynthesis. So for the synthesis of 10 gram of glucose, you need 717.6 kilocalories of the solar energy. Okay, ji. Now we are moving to discuss about the oligosaccharides. So oligosaccharides, the characteristics involved that these are very less sweet in taste. They are less soluble in water. On hydrolysis, they will yield the 2 to 10 monosaccharides. Okay. So if they are made up of two monosaccharides, and they will be known as disaccharides. And if they are yielding, or if they are made up of 3 monosaccharides, so they will be known as trisaccharides, and so on, tetra, penta, hexa, and ultimately will move after 10 monosaccharides, will move to the polysaccharides category. Okay, so the covalent bond that is uniting these 2 monosaccharides together to be get converted into the disaccharides is known as glycosidic bond. ठीक है, तो 2 monosaccharides को आपस में अटैच करने के लिए जो बॉन्ड यहाँ बनेगा उस बॉन्ड को हम कहते हैं glycosidic bond. Okay, so the examples of these oligosaccharides includes the maltose, sucrose, and lactose. All of these sugars are actually disaccharides. Okay, so now about the oligosaccharides, we'll discuss about their structures. Okay, ji. So most familiar disaccharide is the sucrose, and sucrose is made up of the glucose and fructose, and its molecular formula is C12H22O and O11. Okay, so you can see here, this one is the sucrose. Sucrose is formed by the combination of this glucose with this fructose. Okay, so glucose and fructose, you can see here, and the reaction between the 1 and 2. Okay, the reaction between the carbon number 1 and the carbon number 2 of, as you can see here, the carbon number 1 of this glucose and the carbon number 2 of this fructose, they are going to react with each other, releasing a water molecule and forming the glycosidic linkage that is known as 1,2 glycosidic linkage. So this is known as the sucrose. Similarly, you can see here, when glucose is going to combine with glucose, so it will form the maltose. Okay, and in maltose, the on one side the glucose is having carbon number 1, and on the other side the glucose is having carbon number 4. They will react to each other, forming 1,4 glycosidic linkage and converting these 2 glucose into a disaccharide, that is the maltose. Okay, so the polysaccharides. Okay, ji. So polysaccharides are the most complex and the most abundant carbohydrates in nature. They are usually branched and tasteless. So you can see here, these are branched and these are tasteless. They are made up of several monosaccharides linked together by the glycosidic bonds. We can see here, there are a lot of monosaccharides that are linked together by the glycosidic bond. Okay, so they are having higher molecular weights. Okay, and they are only sparingly soluble in water. Okay, they are not too much soluble in water, they are only sparingly soluble in water. So the examples include the starch, glycogen, cellulose, dextrin, agar, pectin, and chitin. One by one, we'll discuss about these examples. So first of all, starch. A starch is found in the fruits, grains, seeds, and tubers. So you can see here, it's found in fruits, grains, seeds, and tubers. Now, it's the main source of carbohydrate for the animals, and it yields glucose, a lot of glucose molecules on hydrolysis. And it has 2 types. The amylose. Amylose is unbranched chain, as you can see here. It's an unbranched chain, and that's why it is easily soluble in hot water. But the second type is amylopectin. You can see here, amylopectin is somewhat branched. Okay, we can see here, amylopectin is somewhat branched, so that's why it is insoluble in hot or cold water. And in laboratory, if you want to identify the starch, then you can done with an iodine test, because starches usually give blue color with the iodine. Okay, so you can see here, the starch is going to be formed when a lot of glucose molecules are being joined together by the alpha 1,4 linkage. Okay, so next is the glycogen. It is also called the animal starch, and it is found abundantly in the liver and the muscles, and it's insoluble in water, and it yield glucose molecules on hydrolysis, and with iodine it will give you a red color, so that you can easily identify it in laboratory. Okay, so you can see here, the glycogen is extensively branched. Okay, so you can see here. Now the cellulose. So students, all of these structures or the types that we are discussing here about the starch, the glycogen, the cellulose, the chitin. So you need to differentiate between all of these types with this with reference to their bonds, with reference to their structures, okay, with reference to their solubilities. So cellulose is the most abundant carbohydrate in nature, and cotton is an example of the cellulose. Okay, and cellulose is also present in the cell walls of the plants, and it is also insoluble in water. Okay, and it yield glucose molecules on hydrolysis. And the most important thing, we cannot digest this cellulose. Okay, because we are lacking the enzyme cellulase that is required for the breakdown of this cellulose. So in herbivores can easily digest the plants containing the cellulose because their digestive tract is inhabiting the microorganisms, the bacteria, yeast, and protozoa that will release the cellulase enzyme there for the digestion of this cellulose. Okay, so cellulose gives no color with iodine. So in this way, you can easily identify it in the laboratory. So you can see here cellulose, and cellulose is unbranched, and you can see here the beta glucoses with one-fold linkage. So beta one-fold linkage exists in cellulose molecules. Okay. Now, the last type is the chitin. Okay, so chitin is used by the arthropods, the insects, specifically because they are having the exoskeleton. So chitin इनके जो है वो exoskeleton का part होता है actually, and fungi is having the chitin in their cell walls. Okay, so chitin is similar to cellulose with beta linkages, except that the glucose monomer of the chitin has a nitrogen containing attachment, as you can see here. मीन इसका जो स्ट्रक्चर है बिल्कुल सेलुलोज की तरह ही है विद बीटा लिंकेज, बीटा वन फोल्ड लिंकेज, बट एक चीज ये है कि इनमें एक नाइट्रोजन कंटेनिंग अटैचमेंट प्रेजेंट होती है काइटिन में. ओके, विद द ग्लूकोज रिंग्स. ओके जी, सो वी हैव डन विद द कार्बोहाइड्रेट्स इन डिटेल. नाउ we'll discuss about the lipids. Okay, so the lipids are a heterogeneous group of the compounds related to fatty acids. Means जो lipids हैं इनमें कोई एक स्पेसिफिक आप कह लें कि जो है वो हमारे पास सब यूनिट नहीं होता. ठीक है, इनके जो सब यूनिट्स होते हैं ना वो डिफरेंट होते हैं. That's why heterogeneous means having different monomers or having different subunits. So they are insoluble in water. These are some properties of the lipids. You need to focus on these properties, that lipids are insoluble in water. Okay, and they are soluble in organic solvents like the ether, alcohol, chloroform, and benzene. Why? Because lipids are also organic in nature, so they really can't get dissolved in the inorganic or water molecules. So it can be easily dissolved in the organic solvents. So examples include the fats, oils, waxes, and cholesterol. Okay. Now, functions of the lipids. Lipid functions. Lipids are the parts of the cellular membranes. Okay. And they are going to store the energy. Lipids are actually storing higher proportion. Lipids are actually storing double amount of energy than the carbohydrates. Why? Because in carbohydrates you have observed that there are a lot of hydroxyl groups, because carbohydrates are hydrated carbons. कार्बोहाइड्रेट्स में जो है बहुत सारे हाइड्रोक्सिल ग्रुप्स प्रेजेंट होते हैं, बट यहाँ पे वो हाइड्रोक्सिल ग्रुप्स कम हैं और कार्बन हाइड्रोजन बॉन्ड्स की प्रोपोर्शन ज्यादा है. दैट्स व्हाई द लिपिड्स आर स्टोरिंग डबल अमाउंट ऑफ एनर्जी देन द कार्बोहाइड्रेट्स. ओके, so lipids provide the insulation against the atmospheric heat and cold because ये impermeable है. lipids को water जो है वो lipids में absorb नहीं हो सकता. तो अगर किसी चीज की कवरिंग हुई है लिपिड से, तो वहां से जो है वो इजीली हीट और कोल्ड या जो है वाटर वगैरह वो डिफ्यूज आउट नहीं हो सकता. ठीक है, so it's a waterproof material, and it serves as a protective layer on the cuticle of epidermis of some plant organs like the leaves, fruits and seeds. तो अगर आप leaves, fruits या seeds को पानी में गिराएं, so we'll observe that they are not going to absorb water क्योंकि इनकी surface के ऊपर जो है वो epidermis की covering होती है, and this epidermis is having the cuticle. So this cuticle is actually made up of lipids, so it will prevent the entry of water into these plant organs. Okay, so all of these are the most important functions of the lipids. Okay, they are the source of energy. Okay, they are providing a maximum amount of energy than the carbohydrates. They are protective as well. Now, the classification of the lipids. Okay, so lipids have been classified as acylglycerols, waxes, phospholipids, sphingolipids, glycolipids, terpenoids, including the carotenoids and steroids. Okay. So now we'll discuss about the acylglycerols. They are composed of the fatty acids and glycerol. Okay. And they are actually the most important acylglycerol is the triacylglycerol, that is also known as the triglyceride or the neutral lipid, because it's having 3 fatty acids and 1 glycerol molecule. Okay, so when these fatty acids and glycerols or alcohols are going to combine with each other, तो इनके दरमियान एक bond बनता है, that is known as ester bond. Okay, and ester is the compound that is produced as a result of the reaction between alcohol and acid. So you can see here, this one is the formation of a triacylglycerol. So you can see here, these are the 3 fatty acids, and this one is the 1 glycerol molecule. And this 1 glycerol and these 3 fatty acids, each fatty acid is releasing 1 hydrogen atom. Okay, because it's an acid, and acids donate the hydrogen atoms. And glycerol, 1 glycerol is releasing 3 hydroxyl ions. So in this way, 3 water molecules will be released, forming the ester bond. So triacylglycerol will be formed here, releasing 3 water molecules. Okay. Now, we need to discuss about the fatty acids, that what are fatty acids and what are their types and what are their characteristics. So, fatty acids contain, in case of lipids, the fatty acids contain even number of carbon atoms from 2 to 30. Okay, and the straight chain attached with hydrogen and having an acidic group. यानी अगर हम fatty acids की बात करें, तो जो फैटी एसिड्स हैं हमारे पास फॉर अगर इनके स्ट्रक्चर को अगर हम देखें, तो इन फैटी एसिड्स में क्या प्रेजेंट होता है जी? फैटी एसिड्स में प्रेजेंट होते हैं 2 to 30 कार्बन एटम्स, ठीक है, और अटैच्ड विद दी हाइड्रोजन एंड हैविंग एन एसिडिक ग्रुप, और इनके साथ एक एसिडिक ग्रुप प्रेजेंट होता है, ठीक है. सो यू कैन सी हियर, this one is the structure of the fatty acid. Okay, this one is the alkyl chain having the carbon atoms, and this one is the carboxylic group. So that's why these are the fatty acids. Now, they may contain no double bond, means they might be saturated, or they might have up to 6 double bonds, means unsaturated fatty acids. So you can see here, this one is the saturated fat. Okay, saturated fat is having saturated fatty acid. You can observe that all of these bonds are the single bonds. Okay, that's why these are saturated. But here you can see that this fatty acid chain is having the double bonds. So that's why these are unsaturated fatty acids. Okay. So animal fatty acids are straight chain, while plant fatty acids are branched or ringed. Okay. And solubility of the fatty acids in melting points, they're going to increase with increase in number of carbon atoms. Okay. So definitely, अगर number of carbon atoms ज़्यादा हैं, तो उन्हें melt करने के लिए भी maximum जो हमारे पास melting points है, वो maximum होना चाहिए, ठीक है. So with increasing number of carbon atoms, you need to increase the amount of heat for their melting. Okay. So palmitic acid is 16 carbon fatty acid. So butyric acid is 4 carbon. So definitely palmitic acid will have much more solubility and will have much more melting point than the butyric acid. Okay. So melting point of this palmitic acid is 63.1, but for butyric acid, it's much less, that is -8 degrees Celsius. And the fats that are containing these unsaturated fatty acids, these are known as these are actually liquid, but the fats that are containing saturated fatty acids, these are solids. So you can see here, the saturated fatty acids, the saturated fats are having the saturated fatty acids with no double bond, so these are solid. at room temperature, but the unsaturated fatty acids or the unsaturated fats are having unsaturated fatty acids with up to 6 double bonds. You can see here a single double bond here. So these are liquid at room temperature. Okay. Animal fats are solid, but plant fats are usually liquids. Fats and oils, both of these are lighter than water with specific gravity of 0.8, and water has the specific gravity of 1. Okay. Now you can see here the formulas of the acetic acid, that is 2 carbon, butyric acid is 4 carbon, palmitic acid is 16 carbon. So you can see here there is no double bond for up to the palmitic acid. Okay, so that's why ये तमाम जो है ये जो है वो unsaturated है, sorry saturated है, ठीक है. But oleic acid, in case of oleic acid, you can observe the double bond between the carbon 9 and carbon 10. So that's why the oleic acid is 18 carbon unsaturated, or you can say mono unsaturated fatty acid, because it's having the one double bond. Okay. Now we're having the waxes. So waxes are mixtures of long chain alkanes with the odd number of carbon atoms from C25 to C35, and alcohols, ketones, and esters of long chain fatty acids. So because we know that the lipids are heterogeneous group of compounds, so you can see here there are almost 3 to 4 subunits or monomers that have been mentioned here for making the waxes. The functions of the waxes, they are protective coatings on the fruits and the leaves. They are present in the insects. Okay, some insects are going to secrete the waxes in their exoskeleton. So they're going to protect the plants from the water loss or any abrasive damage, and they provide the water barrier, means they are water impermeable. Okay, they are working as the waterproof material for insects, birds, and animals like the sheep. Now the phospholipid. Phospholipids are the derivatives of the phosphatidic acid. Okay. Now, what is phosphatidic acid? Phosphatidic acid is made up of actually glycerol, fatty acids, and phosphoric acid. So you can see here, this one is the phosphatidic acid. So initially there was one glycerol. You can see here one glycerol. It got bind with these two fatty acids, and then it got bind with one phosphoric acid. Okay, one glycerol, two fatty acids, and one phosphoric acid. So it's been converted into the phosphatidic acid. अब हमारे पास जो फोस्फोलिपिड्स हैं, दीज आर डेरिवेटिव्स ऑफ फोस्फेटिडिक एसिड. तो अब इस फोस्फेटिडिक एसिड से एक फोस्फोलिपिड कैसे बनेगा जब इसके साथ कोई बेस अटैच होगी. सो यू कैन सी हियर विद दिस फोस्फेट ग्रुप एक बेस अटैच हो गई है, दैट इज नोन एज कोलिन बेस. सो बाय द अटैचमेंट ऑफ दिस बेस it has been known as the phosphatidylcholine. Lecithin means it's a phospholipid now. Okay. So nitrogenous bases like choline, ethanol, amine, or serine, they're going to get combined with these phosphatidic acids to be converted into the phospholipids. So you can see here, this one is the general formula, or you can say physically the structure of the phospholipid. So the phospholipid is having the polar head, okay, that is hydrophilic, water loving, and the non-polar tail. Okay, so non-polar tail is hydrophobic, water repellent. Now let's discuss about the composition of the head and the tail. Okay, so head is having the phosphate group, okay, phosphoric acid. So that's why it's polar. But these two tails that you are observing here, these are fatty acid chains. Okay, two fatty acid chains, one phosphoric acid, and glycerol is present here. Okay, you can see here. And now this one is the choline base. So in the head you can observe the choline base, you can observe the phosphate, and in the neck region you can observe the glycerol, and these are the two fatty acids. Okay, so these are the four components that have been completed for this phospholipid. Glycerol present here. Okay, fatty acid, these are the two chains of fatty acids. Okay, then phosphoric acid that is present in the head, and the last one is choline base that you can also observe in the head. So these are the four parts of this phospholipid. Okay. So phosphatidylcholine is one of the common example that we have seen here. Okay, so the terpenoids are made up of simple units that are known as the isoprenoid units, and the examples include the rubber, carotenoids, steroids, and terpenses. Their functions, they are the major sources of energy. They also play structural role in the membranes of the cells and the organelles, and they provide insulation and mechanical protection and protection from the water loss and abrasive damage. Okay, now we'll discuss about the proteins. So proteins are the most abundant organic compounds that are found in the cells, and they are going to cover about 50% weight of the cell. So the functions, the most important functions of the proteins include that they are performing the structural role in the cell. All enzymes are proteins, and we know that enzymes are going to control your metabolism. Hormones that are going to regulate your metabolic processes, that are responsible for the on and off of these metabolic processes. So hormones are also proteins. Then hemoglobin, this one is the most important protein to which the oxygen is going to bind for being transferred throughout the body along with the blood. Okay. Now the antibodies, they are going to defend your body from the pathogens. They are also proteins. Then we are having some clotting proteins that will prevent the loss of blood from any injury. Okay. Then there are a lot of proteins that are involved in the movement of organs in your body. Okay, as well as that are involved in the process of cell division. So you can see here, some are enzymatic proteins. Okay, these are the enzymatic proteins, and because enzymes are the proteins, actually, they are involved in the chemical reactions. Then the storage proteins, okay, like the ovalbumin. So ovalbumin is present in the eggs. Okay, so storage of the amino acids, and casein, the protein milk, is the major source of amino acids for the baby milks. Okay, and ovalbumin is the protein of the egg white that is used as an amino acid source for the developing embryo. Hormonal proteins. So hormonal proteins are involved in the regulation of these metabolic processes. Okay, contractile and motor proteins like the actin and myosin, they are involved in the contraction of muscles. Defensive proteins like the antibodies, so they are going to get bind with the pathogens to get them destroyed. Transport proteins, they are present in the membranes for the transport of molecules into and out of the cell. Receptor proteins, they are present on the neurons, okay, or on the receptor cells, so that they can recognize these neurotransmitters or these chemical messengers. Okay, then structural proteins that are present in your body structures, like the keratin is present in your hairs, horns, feathers. Okay, and in skin. So now we'll discuss about the subunits of the proteins that are amino acids. So you can see here the structure of the amino acids having a central alpha carbon to which an alkyl group, a hydrogen atom, an amino group, and a carboxyl group is attached. And remember one thing, all the amino acids are being differentiated from each other on the basis of the change of this alkyl group. Okay. So amino acids contain the carbon, nitrogen, oxygen, and hydrogen. So almost there are 3,000 or even more number of amino acids that are present. Okay, so the number of amino acids vary from few to 3,000 or even more. But in our body, about 170 types of amino acids are present in the cells. From these, 25 are involved in making proteins, and 20 are most important amino acids that are making your most important proteins present in the cells. Okay. So the structure of amino acids has been mentioned here that we have discussed. Okay, so if this alkyl group is having the hydrogen, then it will be known as glycine, or if it's having the CH3, then it will be known as alanine. So glycine and alanine are the most simplest, you can say, amino acids. Okay, now the amino acid linkage, that how these amino acids are going to join together. So amino acids are linked together to form the polypeptide proteins, and the linkage between them we can see here. These are the two amino acids, the glycine and the alanine. So the carboxylic group of one amino acid and then amino group of the other amino acid, they are going to react with each other. Okay, so how we can define it? The hydroxyl group of carboxyl group of one amino acid. So hydroxyl group of carboxyl group of one amino acid it's going to react with the hydrogen of amino group of an other amino acid to release a water molecule, making a CN link that is known as the peptide bond. So you can see here glycyl alanine, that is a dipeptide having two amino acids, has been formed here. Okay. The peptide bond will be formed between the amino acids. And if we are going to attach an other amino acid here, so it will be formed tripeptide. If we are going to attach an other amino acid with tripeptide, so it will form tetra and so on. Now the structure of the proteins. Okay, so each protein has the specific properties which are determined by their number, the specific sequence of amino acid, the number of amino acid, the shape of the protein. Okay, so the protein specification is determined by the amino acid number and the sequence as well as the shape. So there are following levels of the organization of the protein. The primary structure is just a linear structure of amino acids. Amino acids are just attached one after the other in a linear way. Okay. And the F. Sanger was the first scientist who determined the sequence of amino acids in a protein, and the protein was insulin protein. He said that insulin is made up of 51 amino acids arranged in two chains. One is having 21 and the other is having 30 amino acids. And these two chains are held together by the disulfide bond. Okay. Then hemoglobin, it was the structure of the hemoglobin or the sequence was determined by the Vernon Ingram. So he said that there are four chains of the hemoglobin, two alpha and two beta. Each alpha chain is having 141 and each beta chain is having 146 amino acids. Okay. So there are total 574 amino acids in hemoglobin. Now you can see here about 10,000 proteins are present in a human body, which are made up of these 20 amino acids. And the sequence of the nucleotides is very much important for making these specific amino acids. If in the DNA, only one nucleotide is not in its normal position, then the amino acid, the amino acid that is going to attach, it will be a wrong amino acid. So it will surely have some effect on the function of such protein. So you can see here, the disturbance in the primary structure of the proteins you can observe here is the name of this disease that is known as the sickle cell hemoglobin. Okay, so this one is a normal hemoglobin. You can see here from 1 to 7, the amino acids have been arranged. But if the nucleotide in the DNA is disturbed, so you can see here at the sixth position, if the nucleotide is disturbed, okay, if one nucleotide is not in its normal position, so in place of glutamic acid, you will observe a wrong amino acid that is valine. So that's why normally, due to this hemoglobin, the red blood cells are the red blood cells are biconcave shaped. But if this amino acid is not being synthesized properly, then you will observe a sickle cell red blood cell. Okay, so it will not carry the oxygen properly in the body, so you will get anemic by the process of sickle cell hemoglobin disease. Okay, now the secondary structure. So the polypeptide chain coil into a helix. So you can see here, the primary chain hai, it's going to be coiled into the helix in the helical structure. Okay, and this one is known as alpha helix. Now, beta pleated sheet. This one is the beta pleated sheet that is going to be formed by the folding back of this alpha helix. So in alpha helix, there is the geometric structure with 3.6 amino acids in each turn. Okay, 1, 2, and 3. 3.6 amino acids are present in each turn of alpha helix, and beta pleated sheet is formed by the folding back of this polypeptide or this alpha helix structure. And this structure is held together by the hydrogen bonds, as we can see here. The alpha helix is maintained by these hydrogen bonds together. Okay. Now the tertiary structure. So polypeptide chain bands unfolds upon itself, forming the globular structure. Okay, and these proteins are known as the tertiary proteins, and there are three types of the ions that are present in the tertiary structure. The ionic, hydrogen, and disulfide bonds are present here. Okay, and the hydrophobic amino acids are present inside, but hydrophilic amino acids are present or arranged outside this tertiary structure. Now, the quaternary structure. So, polypeptide tertiary chains are aggregated and held together by the hydrophobic interactions, the hydrogen and the ionic bonds. Okay. So, quaternary structure में simply ये होता है कि जो बहुत सारी tertiary structures हैं, ठीक है, जो tertiary form में arrange हो चुकी हैं, they are going to combine with each other forming the quaternary structure. So, quaternary structure जो है वो एक से ज्यादा तरह की जो chains हैं उनका mixture होता है. So here is an example of the hemoglobin that is having two alpha and two beta chains. So you can see here कि इनकी जो chains हैं they have been arranged in the form of tertiary structures. And these are held together by the hydrophobic, hydrogen and ionic bonds. Okay, ji. Now we'll discuss about the classification of proteins. So the proteins have been classified into the following types, the fibrous proteins. So fibrous proteins are having one or more polypeptides in the form of fibrils. They are having secondary structure. They are insoluble in aqueous medium, non-crystalline and elastic in nature. They are performing structural roles in the body. Example include the silk fiber that we are obtaining from the silkworm, myosin that is present in your muscles, fibrin that is present in your blood clot, and keratin that is present in your nails and hairs. So that's why it's having the secondary structure and it's performing the structural roles in your body. Now we are having the globular proteins. So these are spherical or ellipsoidal in shape. They are having tertiary structure. They are soluble in aqueous medium. They can be crystallized. Okay. And examples include the enzymes, antibodies, hormones, and hemoglobin. So all of these are the globular proteins that are present in your body. Okay. Now we'll discuss about the nucleic acids. Okay. So nucleic acids, which include the DNA and RNA. Okay, so the nucleic acids, DNA and RNA discovery. So who discovered the nucleic acids? They were discovered by the Frederick Miescher in 1869 from the nuclei of the pus cells. Okay, and how they were isolated. When they were isolated, they were named as nucleic acids because nucleic acids were discovered from the nucleus, and because of their acidic nature, they were named as nucleic acids. Okay. Now, what are the types of these nucleic acids? There are two types. The first one is the oxyribonucleic acid or DNA, and the second one is the ribonucleic acid or RNA. Okay. Now, DNA occurs in the chromosomes, in the nuclei of the cells, in the mitochondria and chloroplasts. But RNA is present in the nucleolus, okay, and in the ribosomes and in the cytosol, okay. Now, nucleic acid subunits. So the subunits are the nucleotides. And this nucleotide has the three subunits. The first one is the 5-carbon monosaccharide sugar, as you can see here. Okay. Now, second one is nitrogen containing base that is attached to the carbon number 1. This one, the nitrogen containing base that is attached to the carbon number 1 of this 5-carbon sugar. Okay. Now, the third is the phosphoric acid that is attached to the carbon number 5. Okay. So one sugar and then the nitrogenous base that is attached to carbon number 1 and the phosphoric acid that is attached to carbon number 5. Okay. Nitrogenous bases are of two types. So the pyrimidines. Pyrimidines are the small and single ringed, and these are cytosine, thiamine, uracil. Okay, and cytosine, thiamine, or uracil. And purines. Purines are large and double ringed, and they include the adenine and the guanine. Okay. Now, the phosphoric acid has the ability to develop the ester linkage with hydroxyl group. And the compound formed by the combination of base and pentose sugar is known as the nucleoside. Okay, means the phosphoric acid. This phosphoric acid has the ability to make bond with the hydroxyl group of the carbon number 3 of the other nucleotide. Okay, so when base is going to make a bond with the pentose sugar, when this pentose sugar is having attached nitrogenous base, so this structure will be known as the nucleoside. And when this nucleoside will get attached to the phosphoric acid, so it will be converted into nucleotide. तो न्यूक्लियोसाइड और न्यूक्लियोटाइड का डिफरेंस यहाँ पे आपने देखना है. न्यूक्लियोसाइड इज द 5 carbon sugar with base, and when it's going to attach with phosphoric acid, it will be converted into a nucleotide. So if there is only one phosphate, so it will be mono. If there are two phosphate, it will be di, and if there are three phosphates, so it will be tri. Okay. Now, the differences between the DNA and RNA. So DNA is deoxyribonucleic acid, and RNA is ribonucleic acid. Okay, these are the two. The DNA is having deoxyribonucleotides. Okay, but RNA is having ribonucleotide. These are their subunits. Now, the DNA is having pentose sugar, that is deoxyribose sugar, in which one oxygen is being removed from the hydroxyl group at the carbon number 2. But in case of RNA, the sugar is ribose sugar. Okay. And in case of DNA, the nucleotides include the adenine, thymine, cytosine, and guanine. These are the bases. But in case of RNA, the bases include the uracil. All three are same, but the thymine will be replaced by the uracil in case of RNA. Okay. Now the last difference is that DNA is the double-stranded molecule, but RNA is single-stranded molecule. Okay. DNA, deoxyribonucleic acid. So, DNA is hereditary material, and it controls the properties and activities of the cell. So, there are 4 types of the nucleotides in the DNA. D-adenosine monophosphate. D stands for deoxy. Okay. D-guanosine monophosphate, D-cytidine, and D-thymidine monophosphate. Okay. So, you can see here in case of DNA, the nitrogenous base, when it's going to attach with the deoxyribose sugar, so it will be converted into a nucleoside. Okay. After that, when it will get attached with phosphoric acid, it will be converted into a nucleotide. So these are the names of these nucleotides mentioned here. Okay. So these nucleotides are united with one another through the phosphodiester linkage. Okay. And when two will be attached together, it will be a diphosphodiester linkage. And so when a lot of nucleotides will be attached together, so a polynucleotide chain will be formed. Okay. NAD, nicotine, a nicotine amide adenine dinucleotide. So it's a coenzyme. Okay. 1951, the Erwin Chertoff said that the ratios of the bases are equal in this DNA molecule, and the adenine and thymine are equal in ratio, just like the guanine and cytosine. So then Maurice Wilkins and Rosalind Franklin, they provided the technique of X-ray diffraction, and after that James T. Watson and Francis Crick, they developed the scale model of DNA. So there are some points related to scale model of DNA, that the DNA is having two polynucleotide strands that are anti-parallel to each other. Okay, they are in the form of a double helical structure. Okay, and they are anti-parallel to each other. One is 5 to 3 and other is 3 to 5. And after that, the two chains are held together by the weak bonds. Okay, hydrogen bonds between the base pairs. So the adenine is going to make 2 hydrogen bonds with thymine, and guanine is going to make 3 hydrogen bonds with the cytosine. Okay. So the two strands are wound around each other so that there are 10 base pairs in each turn. So in each turn, there are 10 base pairs. Okay. And the one turn has a diameter of about 34 angstroms. Okay. So DNA, the amount of DNA is fixed in particular species, and in every cell, the amount of DNA is same, but in the germ cells, it's going to be half. As you can see here, in chicken, the amount of DNA, in carp, the amount of DNA. So in the normal cells and in the germ cells, you can observe a clear-cut difference of about half DNA, okay? Now the RNA, that is a ribonucleic acid. So the RNA molecule occur in the single strand, and it might fold it back to be converted into a double helical model. So it's having the 4 nitrogenous bases: the cytosine, guanine, the uracil, and the adenine. And the RNA is going to be synthesized from the DNA by the process of transcription. So there are following three types of RNA: messenger RNA, transfer RNA, ribosomal RNA. Okay, and they are going to be synthesized in the DNA, in the nucleus from the DNA by the process of transcription. So first of all, the messenger RNA. So you can see here it's going to be synthesized in the nucleus from the DNA. So on the DNA, one gene is going to be copied in the form of or transcribed in the form of messenger RNA. So it will carry the information of one gene from this DNA to the cytoplasm. Okay, and the length of this messenger RNA depends upon the length of the gene. Okay, and if we are having the messenger RNA with 3000 nucleotides, then we'll have the protein with 1000 amino acids, because we know that three nucleotides will form a codon, and one codon specifies one amino acid. So the messenger RNA is present about 3 to 4% in the cell. Now the transfer RNA, it picks up the amino acids when this messenger RNA is going to move in the cytoplasm. So now transfer RNA will read the message on this messenger RNA, and it will transfer the specific amino acids on this ribosome. Okay, so transfer RNA, the name shows that it will just transfer the amino acids, the specific amino acids, the ribosomes, for being converted into proteins. Okay, so there is one specific transfer RNA for each amino acid. And we know that our cells have 20 kinds of transfer RNA for 20 kinds of specific amino acids. Okay. The length of transfer RNA is about 75 to 90 nucleotides, and the percentage in the cell is about 10 to 20%. Now, the ribosomal RNA is a machinery for the synthesis of proteins. First of all, ribosomal RNA will make the small and larger subunits of the ribosomes, and then these subunits will get joined together to make the ribosome. Now, ribosome is providing the site where these amino acids will be dropped and where the peptide bonds will be formed for making the proteins. Okay. So about 20%, about 80% of the cellular RNA is the ribosomal RNA. Now, the conjugated molecules, when two different molecules belonging to different categories combine together, they will form the conjugated molecules. Okay. So when carbohydrates will combine with proteins, they will form glycoproteins, and when with lipids, they will form glycolipids. Okay. The glycoproteins are actually, the cellular secretions are actually glycoproteinic in nature. Okay, okay. And glycoprotein and glycolipids are also the structural components of the plasma membranes. Lipoproteins are formed by the combination of the lipids and the proteins. They will also form the framework of all types of membranes. Nucleic acids are also in the form of these conjugated molecules and the form of nucleoproteins. In the DNA or in the RNA, the DNA is going to be combined with the histone proteins to be converted into the nucleoproteins. Okay. So they are involved in the regulation of gene expression. Now we'll discuss about the enzymes. So the enzymes are biologically active proteins. They are tremendously increasing the efficiency of a biochemical reaction. Means they're going to increase the efficiency or increase the speed of a biochemical reaction. Okay, they are not going to on or off the biochemical reaction. For these purposes, the hormones are there. Now the enzymes composition. So enzymes are made up of hundreds of amino acids joined together and coiled upon themselves to form the globular structure. Now the enzyme structure. So the catalytic activity of the enzyme is restricted to a small portion of the structure that is known as active site, as you can see here. This one is the entire enzyme. This enzyme is having this active site. It's a site to which the substrate will be attached and will be converted into the products. Okay. So catalytic activity is the type of activity in which substrate will be converted into its product. So an enzyme and its substrate will react with each other through a definite charge bearing site of enzyme that is known as active site. It's an alternate definition of the active site. Okay. Now, the reactant is called the substrate. It has to attach at the active site. Now, active site is further divided into two regions. The first region is binding and the second is catalytic. So, binding name shows that this site is only responsible for the proper attachment of the substrate. And after the attachment of the substrate with this binding site, it will activate the catalytic site, and now catalytic site is responsible for the conversion of this attached substrate into its product. Okay. Now the cofactors and its types. Okay. So mostly the enzymes have the are actually the proteinic in nature, and these enzymes have the complete structure that is proteinic in nature. But some enzymes have non-protein parts that are known as cofactors. Okay, and what are the properties of these cofactors? They are essential for the proper functioning of enzymes. They act as bridge between the enzyme and its substrate. Okay, and they are the source of chemical energy. Okay, and now types of the cofactors. If the cofactor is inorganic ion and it is detachable, means we can detach it, so it will be known as activator. And examples of activators include the metal ions, as you can see here. Okay, and if the cofactor or the non-protein part is covalently bonded to the enzyme, it will be known as prosthetic group. But if it is loosely attached to the protein part, it will be known as coenzyme. Okay. So examples of coenzymes include the vitamins. So we have studied about that. That cofactor is the non-protein part. If it is covalently bonded to enzyme, it will be a prosthetic group. But if it is loosely attached, then it will be a coenzyme. Okay, and this one, it will be known as activator if it is inorganic ion and it is detachable. Okay, it can be detached because it's making weak bond with the enzyme. So a cofactor is required for the proper functioning of enzyme. It acts as a bridge between the enzyme and substrate. It's a source of chemical energy. So these are the properties that are very much important related to the cofactors. Okay, so you can see here in the previous diagram, the cofactor that has been mentioned here. Just forgot to put our diagram. So this one is the cofactor molecule. Okay, so you can see here, the substrate will not get bind to this active site until and unless this cofactor is not attached to it. So when the cofactor will get attached, then the substrate will be attached to the enzyme. So now it is responsible for the catalytic activity for being converted into proteins. Okay, so we have done with this part. I just moved to the previous slide for the diagram. Okay, so an enzyme with its coenzyme or prosthetic group removed is known as apoenzyme. Means you can see here, this enzyme is having not any attached cofactor, so that's why it will be known as the apoenzyme. Apo means away. Away enzyme means one enzyme is away from its prosthetic group or its coenzyme, or you can say its cofactor. But if an enzyme has attached cofactor, okay, if an enzyme has attached cofactor, or you can say coenzyme or prosthetic group, that are the types of the cofactors, so it will be known as holoenzyme. Holo means complete or activated enzyme. Okay. Now, the location of enzymes. Enzymes are present in the cell's cytoplasm as well as in certain organelles. So, enzymes responsible for the photosynthesis are present in chloroplast. For the respiration are found in mitochondria. For the synthesis of protein are present on ribosomes. Okay. Now, we'll discuss about the characteristics of enzymes that are very much important. So all enzymes are globular proteins, okay? And they are going to increase the rate of reaction without themselves being used up. They will be not utilized. Means they are not going to be broken down, and they just need to attach with the substrates to convert them into the products. Okay. So their presence does not affect the nature or the properties of end products, and small amount of the enzyme can accelerate the chemical reaction, and they are very specific in their action. A single enzyme can catalyze a specific substrate into a product. Okay, same enzyme cannot be used for all of the substrates. So they are very sensitive. They might be destroyed or they might have some complexity in their functioning if you're going to change the pH, temperature, and substrate concentration. Okay. So Enzymes require a cofactor for their proper functioning, and they lower the activation energy of the reactions. Okay. Now the mechanism of enzyme action. So enzymes, by virtue of its specificity, recognizes and reacts with a special chemical substance called the substrate. Okay. So first of all, enzymes are going to react with the substrates. Okay, so when enzyme will react with substrate, enzyme substrate complex will be formed, and then the products will be formed and separated. Now, models for enzyme working, according to Lock and Key model, the that was proposed by the Emil Fischer in 1890. He says that the enzyme's active site is rigid structure. Okay, so if a specific substrate binds to this active site, then it must have a specific shape to get attached to the enzyme's active site. And after that, induced fit model by the Koshland. It explains that the enzyme's active site is having some kind of flexibility. So if a substrate will bind to it, it will be get modified. Okay. So now factors affecting the enzyme action, the enzyme concentration. So about the enzyme concentration, the rate of the reaction depends directly on the enzyme present. If you're going to increase the enzyme concentration, the rate of reaction will be doubled, but it is up to a certain limit. Okay. And the substrate concentration will also have effect on the rate of enzyme action. If you are going to increase the substrate availability, the present enzymes will increase the rate of reaction. But after that, you need to add more enzymes for increasing the rate of reaction. Okay. And then temperature. So temperature, the rate of reaction may increase with increase in temperature, but also up to a certain limit, because all enzymes have optimum temperature. And after the optimum temperature, if we are going to increase the temperature, then the enzymes are going to denature. So for the human body, the optimum temperature is 37, because heat is a source of activation energy and kinetic energy. So more heat will cause the collisions of these reactants and the enzymes together, so that they can denature the enzymes. Okay. And after that, pH value. So if you're going to change the pH of the enzyme, then it will change the ionization of the amino acids. Okay. And the change in pH will change the ionization of the amino acids on the active side. So definitely specific substrate cannot bind to it. Okay. And after that, we'll discuss about the inhibitors. So inhibitors are some kind of chemicals or the poisons or the substances that can react in place of substrate with enzyme. Okay. They can block the active site permanently or temporarily. So examples include the cyanide, antibiotics, antimetabolites, and some drugs. So there are two main types, irreversible and reversible. So irreversible are those that are going to make the weak bonds, okay, I'm sorry, covalent bonds with the active site, and they're going to destroy the active site or block the active site permanently. Reversible are those that are going to make the weak linkages with the enzyme, and their effect can be neutralized if you are going to increase the concentration of substrate. Okay. And then there are two major types of the reversible inhibitors, the competitive and non-competitive. So competitive are those that are having structural similarity with substrate. So definitely an enzyme will recognize it, but it will not convert this competitive inhibitor into the product. Okay. And after that, non-competitive inhibitors, so they are not having any structural similarity with the substrate. So they will bind to at an other site, and if they are going to bind with at an other site on the enzyme, so they will change the structure of enzyme in such a way that if a genuine substrate will try to bind with the enzyme, so it's not going to bind with that enzyme to being converted into the product. Okay. So we have done with this, and inshallah we'll meet up with the next video. Until then, Allah Hafiz.